Well, hello everyone, and welcome to today's webinar from 1 Million Solutions in Health. My name is Dawn Van Dam, and I am the President and CEO of 1 Million Solutions in Health. We're extremely excited to be having today's webinar because this is the kickoff of our brand new partnership with the National Institutes of Health Office of Technology Transfer and the NIH National Cancer Institute. So we're going to tell you a little bit about how this is going to work and hopefully get lots of your feedback and hopefully tell you a lot about this great science and technology. So this is what's called a Signature Square program, and I will go through that with you just in another moment. So we're going to go through some welcome and introductions, so information that I'm going to provide you about the process. And then Dr. Peng Lo will give her presentation. She is from the NIH. Then we'll talk about getting your feedback on evaluating the information that's been presented, do some closing remarks, and ask you to complete our survey. Now, our survey is not just a typical survey of, give us some information about what you think about this webinar. It's really an extremely important part of this process in that the Signature Square is a way for end users, industry, academia, other organizations to help evaluate this science and its applications into the marketplace. So this is one of the key reasons why the NIH has partnered with us at 1 Million Solutions in Health, because we have this tool called the Signature Square, which can help them get feedback and help them have information on the roadmap for this future technology, or for this current technology and its future applications. So a little bit about 1 Million Solutions in Health. Our goal is to help improve healthcare one solution at a time. So today we're talking about one solution and one application, but we have lots of different things that we offer throughout the year in life sciences, healthcare, and for patients and consumers. And our goal is to shape healthcare by sharing solutions. Too many times new science gets put into the back of the train, so to speak, because it either doesn't get the right feedback or it doesn't get the right funding. So a little bit about 1 Million Solutions in Health. We're here to help people connect, to help you learn and share, and to innovate. So if you have some ideas that you'd like to share, whether you yourself are in academia, government, you're an entrepreneur, you're in business, we can work with you to see how we can share that with our very large network. We have a database of over 5 million people all across the world in every discipline. And of course, we like to help people collaborate, and one of the ways that we do that is through our Technology Evaluation Consortium, which I'll just give you a few highlights about in a moment. So today's presentation, let me just give you a brief overview about that. The title is Testing the Efficacy of New Cancer Treatments in Clinical Trials Using a Unique Biomarker. So just to give you the background, 1 Million Solutions in Health has established a one-of-a-kind new partnership with the United States National Institutes of Health Office of Technology Transfer and the National Cancer Institute Technology Transfer Center. And the goal there is to increase awareness and understanding of NIH technologies by potential partners in the life science and healthcare industries. Part of that process is this very unique partnership with the NIH Office of Technology Transfer, which allows us to present novel ideas, innovation, or research to you with an aim to increase the partnering and/or licensing opportunities for NIH-developed technologies. And the goal there is to increase the technology's visibility within the life sciences and healthcare industries. And really the idea there is this will serve to further support research, product development, commercialization, and economic development opportunities. The NIH is going through a lot of changes right now, and this is one of those very positive changes which will come with the rest that are going to be announced over the next while through their organization. But here's a chance to move beyond, as they say, beyond their Rolodex out to our database of 5 million people. And this first NIH opportunity will really showcase how a new biomarker technology uniquely measures the progression and aggressiveness of the patient's cancer. And this biomarker also predicts recurrence and metastases in various cancers, including liver, colorectal, adrenal, and lung cancer. So as I said, we're very excited to be bringing this to you today. So just a tiny bit about the Technology Evaluation Consortium. Through 1 Million Solutions in Health, we have various technology evaluation consortiums. In this one here, it's a collaborative partnership within the pharmaceutical and biotech industry and other stakeholders like the NIH. We're also trying to reach out to other governments around the world and other jurisdictions to help bring their technologies forward as well. Our ultimate mission is to be the place for the industry and academia and government, it should say there, to conduct their evaluations and validations. So today we're going to do an evaluation, and through a more intensive project process, we do validations. And really, the other part of our mission is to triage and streamline new technology consideration for the industry and to help things get to commercialization faster. That's our huge, huge frustration point. So as I said, we bring together the pharmaceutical and biotech industry or other end users. And then on the other side, we bring together the technology vendor or scientist through the Technology Evaluation Consortium in order to get feedback. Here's just a sampling of some of our current members or past members. You may recognize some of these people from the industry. Some of their pictures may be current, some of their pictures might be old, but you can see that it's an established high-level group of people who constantly is giving us feedback on what new technologies should and could be adopted into the pharmaceutical industry and biotech industry. So really what our process is very, very simple. We find new scientific solutions. We gather input to optimize those solutions either through a program like today's Signature Square or through our TEC validation projects. And then we share the findings with the 1 Million Solutions in Health community. Today, you're lucky enough to be joining us to hear this firsthand from Dr. Lowe, and we're really happy to have her. So just a moment about why the National Institutes of Health, and in particular their Office of Technology Transfer, sees this as making sense. Well, first of all, for the industry, it's a view into the work going on at the NIH and the roadmap for the various technologies that we'll be profiling. It's a chance to have you ask some questions and get some answers, and just in case you haven't figured it out yet, you just stick your questions into that chat box down the bottom, send it to us, and I will ask our speaker those questions on your behalf. And then through the Signature Square survey, we're going to ask you to give some intensive evaluation. Now this is really, really critically important, as you can see here to the NIH, is they want feedback, feedback, feedback through this process of expanding their Rolodex through our consortium and our database, because they really want to get some value out of the science that is being developed and patented and find opportunities for licensing agreements, find opportunities for CRADAs, find opportunities for people to partner with scientists like Dr. Lowe to bring this technology further to market. So what you're going to see in all of these NIH Signature Square presentations is not fully ready to be utilized tomorrow, but with your help and your participation and perhaps your company taking on this science, it can then be a win-win for all participants, including more technology adoption. So today is the first kickoff of the NIH events, and I'm just going to tell you a little bit about the Signature Square program. I'm going to ask my partner here at 1 Million Solutions in Health, Adiola, to just put the Signature Square survey up in the chat box for everyone. So I would ask you to go to that survey now and open it during the webinar, because really it's your chance to provide feedback as we're going through the presentation. You're going to give us a scientific evaluation and feedback of the technology. You're going to listen to the presentation, and you're going to actually rate the presentation, not the presentation, but the science and its applicability in a number of different areas. So as I said, we're going to have you go through the webinar. You're going to give us some evaluation, and you're going to do it in two areas. One is, how ready is this technology to be utilized by the world? And then what's its value to the marketplace? So it might not be all that ready today, and that's where you come in and you could help out, but it might have huge applications in the marketplace, and that's, again, where we want your feedback. After that, we will produce a summary report that will go to the scientist and the NIH in this case, outlining the scores and the qualitative feedback provided by the respondents. And all of this you'll be able to do anonymously. However, we would also love you to give me a call afterwards if you're interested in talking to Dr. Lowe and her colleagues in the Office of Technology Transfer about partnering with her to take this science forward. So those are our two key metrics. And our success factors is you filling out the survey and then you giving me a call or sending me an email saying that you want to get involved in helping move this forward. In the end, the results will be put onto our signature square. Is this a blockbuster opportunity, an upstart novel, or an innovator? So that's how it works. And the benefits, this will help the industry by increasing the flow of technologies being evaluated. We know that everyone's struggling to be more efficient and more effective, and this is one of the ways to help things out. And it provides everyone out there listening with the opportunity to learn more about different scientific technologies and expedite the consideration and hopefully the adoption of these technologies, because it will accelerate that feedback loop between the industry and the technology provider, in this case, Dr. Lowe and her team. The benefits for the NIH, I've kind of mentioned some of these already, but they're hoping to have more licensing agreements coming out of this program, to have more creatives. And if you don't know what a creative is, I'll just let you look that up. But it's basically a partnering agreement to help develop the product further, or the technology or the science further. And then they want more feedback and direction for their roadmap. So again, that's why it's so important that you fill out that survey. So I hope by now everyone's opened up that survey link and started to take a look at it. So just a little bit more about what Dr. Lowe is going to present, and then we'll move on. So as I said, this is your opportunity to participate in this exclusive signature square process where the end users rate and evaluate the technology, provide feedback directly to the scientists, and help affect and drive the NIH research roadmap for this technology. If you're here, you probably read the email, and the email said that an important application of this particular biomarker would be its use in clinical trials to better evaluate each patient as to the aggressiveness or progression of their cancer. And this is really important because this biomarker has shown that some patients who are traditionally categorized as things like stage 1 cancer are actually harboring an underlying disease which is much more aggressive than expected. So the application of this biomarker in clinical trials could serve to potentially revolutionize oncology clinical trials, which is why we're so excited about it. So let me give you just a quick background about Dr. Lowe, and then I will turn things over to her to deliver the presentation for today. So Dr. Lowe's biography. She is the head of the section on cellular neurobiology, the program on developmental neuroscience at NICHD. Dr. Lowe received her Bachelor of Science, her honors Bachelor of Science in biochemistry from University College in Dublin, Ireland, and her PhD in molecular biology from actually my alma mater, the University of Pennsylvania in Philadelphia, Pennsylvania in the United States. She did her postdoctoral work at the NIH and at the Max Planck Institute in Germany. During her career at the NIH, Dr. Lowe has received numerous honors and awards, including the Public Health Superior Service Award, the NIH Directors Award, the NIH Directors Award for Mentoring, the FASEB Award for Excellence in Science, and the Women in Endocrinology Mentor Award. She is a member of the Endocrinology Society, American Society for Cell Biology, the Society for Neuroscience, and served on many editorial boards. She has authored more than 160 scientific articles published in the most prestigious journals such as Cell and Nature Medicine. So with no further ado, I'm going to pass the ball over to Dr. Lowe, let her unmute herself, and let her start with her presentation for today and answer your questions that you're going to stick into the chat box to me, and I will moderate those questions as we go through. So over to you, Dr. Lowe.
Thank you very much, Dawn, for the kind introduction. So good morning to everyone who is attending, and I'm very grateful and thank you for joining this webinar to share with you our discovery of a new biomarker called carboxypeptidase E, and the fact that this biomarker can be used for screening and predicting recurrence and evaluating treatment efficacy of the cancer. So I shall begin by saying that predicting recurrence of cancer will definitely increase survival. The marker can be used for early detection of cancer, which will improve outcome, and evaluating the efficacy of cancer treatment in drug development would be a major advantage to the therapy of cancer. So to begin with, what is carboxypeptidase E? It's actually a gene with 9 exons that has been found to be a processing enzyme for prohormones, but we found that it is highly expressed in metastatic cancer cells. The two forms of message, one is the wild type, and the other is a splice variant form. And for today, the reference would be just to CPE biomarker. It can contain both of these forms, the wild type and the splice variant, and together it can predict future metastasis of the patient. So here's an example of many types of cancer cell lines that we have looked at, where we have assayed for the CPE biomarker. And the ones in red are the highly metastatic cell lines, and the ones in blue are the low metastatic cell lines of the same type of cancer. For example, HCC is hepatocellular carcinoma, and the cell lines that are highly metastatic have high levels of the biomarker. And again, in breast cancer, ones that are highly metastatic have high levels, and likewise in colon and head and neck cancers. So there's a good correlation between metastasis and this biomarker. So this is an example where we can downregulate the expression of this biomarker, the CPE gene, and we did that for liver cancer cells, and then implanted those cells into the liver of nude mice. And on the left panel, you can see that we have control mouse in which we have just treated the cells with scrambled SI or SH RNA, where it won't downregulate the gene, but on the right mouse, it has been downregulated in expression of CPE. We wait 35 days, and you can see that the control mouse that still expresses CPE biomarker has grown a huge tumor and also metastasized to the lung and the kidneys and the intestines. And on the right, you can see that the mouse that has been suppressed in expressing this biomarker does not have a big tumor nor show any metastasis. So this gene is clearly really important for controlling metastasis of tumors. So at the clinical level, then we took this from the basic science to the clinical level, and we found that 30% of early stage 1 and stage 2 cancer patients, and we have a study that has been completed for liver and for lung patients, they have very high levels of this biomarker. And 80% of the 30% showed recurrence or spread of the cancer within 2 years. And currently, most doctors wait until symptoms recur before taking further action with these stage 1 and stage 2 patients who are considered low risk based on the histopathology, and this is what they've got to go by, is the histopathology. So this came as a great surprise that there were as much as 30% of the stage 1, stage 2 patients actually had high levels and showed recurrence. So here's the Kaplan-Meier plots that we have done in an example. Just for example, we did it on lung cancer patients, stage 1 and stage 2, and we determined a cutoff of 10,000 copy numbers of the biomarker as ones that would actually, ones that are above will show rapid recurrence, as shown in the dotted line on the left graph. You can see that the patients with copy numbers greater than 10,000 showed a rapid recurrence rate compared to the black line where they had copy numbers less than 10,000. And if you go to the right graph, and this is a prediction of survival or death, and you can see that the dotted line is the one with greater than 10,000, and those patients showed rapid death versus the patients that have less than 10,000. So in this way, we're able to detect early detection of patients that actually show, or we can predict that they can show us when they're going to show recurrence, and that's really important. So just taking a look at some of the other analysis that we have done with this group of patients, We see that recurrence is, of course, highly significant in terms of the p-value and also in terms of death. And when we looked at the smoking profile, it was interesting that the non-smokers showed a greater death rate than we expected, actually, and as shown on the right side, where there's a star 0.28. And with terms of age, patients that were greater than 60 years of age showed a significant recurrence. And with regard to gender, it's also interesting that the females, and actually most of these females were non-smokers, had a higher death rate when the biomarker is high. So the biomarker then in these resected tumors will identify early stage 1 and 2 cancer patients that are at high risk of recurrence within a short time when high levels of this biomarker are in the tumor. This will then prompt a proactive approach post-surgery with frequent follow-up for early detection of first signs of recurrence of spreading. And it will also indicate need for aggressive treatment post-surgery to prevent recurrence and spread of the cancer. And as I had said before, most of these patients in stage 1 and stage 2 are given good prognosis before, based on their histopathology. So being able to catch this group of patients with aggressive tumors at stage 1 and stage 2 will greatly increase survival rate. So now I'm going to turn to the progression of our research, which went from analyzing frozen resected tumors, where we would first identify all the different tumors that express this biomarker. We then wanted to develop a blood assay, and this is something is well known in the field called liquid biopsy. It's a very exciting progression of being able to actually determine the biomarker in exosomes, and we have found that exosomes that in the circulating in the blood contain CPE mRNA, and we can use that in the diagnosis of cancer. So the serum plasma assay we use, we measure the CPE or biomarker levels, and we show that it is elevated in exosomes of cancer patients compared to normal subjects. And while we haven't any data right now, we expect that the levels of the biomarker to decrease significantly when patient becomes cancer-free post-surgery. And this next slide just gives you the clinical data that we have obtained so far. We have 27 controls that are normal volunteers, and we have 33 patients that have pheochromocytoma paraganglioma tumors. We have those patients at NIH, and we were able to get the patients early, get their blood and run over to the lab and make the exosomes and do our assay. And the advantage of having the patients at the NIH is that we're following them right now. Before surgery, we would take a sample, and then after surgery, we are collecting from these patients again to re-assay. To do the second part of what we wanted to do was to look and see if the biomarker goes down, which we expect to do so probably around 3 to 6 months after their surgery. So just a little bit more about these patients. We able to show that the significant difference of a p-value of 0.008 and 0.001 in two different types of statistical analysis. So it's highly significant in terms of distinguishing the group that are normal and the group that are have cancer in terms of the levels of this biomarker. We also show in this slide on the top that the age does not matter. There's no difference in p-value between the different ages, and there is no difference in males or females either. So it's a pretty general in terms of being able to catch patients with this biomarker who have, in fact, cancer. So what are some of the advantages? So this biomarker then is expressed in many types of tumors, and we've done it and found that it is expressed in lung, liver, kidney, adrenal, colon, thyroid, ovarian cancers, and also, and more recently, we found it in glioma patients as well. And the serum then can be collected and assayed for this biomarker. The assay is non-invasive and, of course, less costly than biopsies. So how can we use this biomarker in terms of being able to assay in blood? So routine screening of populations of greater than 60 years of age, simple tests to determine whether there's a possibility that one has a tumor and remains undetected before symptoms arise, and by looking at the biomarker, one can follow it up if the person is high to other analysis. So secondly is the screening of high-risk patients for early detection of many types of cancers, including patients with liver cirrhosis, smokers, men with positive PSA tests, women with solid ovarian cysts. There's no way except biopsy right now to look at those patients, and also the ones that carry the gene for breast cancer. Those are high-risk group, again, that could be screened. And the pheochromocytoma patients with inherited gene mutations for the disease, those patients can be followed up to determine when they actually first get a tumor. Post-surgery, screening of stage 1 and stage 2 patients for early detection of recurrence would be very useful, and certainly in clinical trials to test the efficacy of the cancer treatment will be a major goal in this type of assay in order to follow a patient with various types of therapies that they may be getting. So there's a huge market opportunity. In liver cancer, patients infected with hepatitis B or C, those numbers are increasing both in America and also in the rest of the world. Patients that have cirrhosis from alcohol or drug abuse, another high-risk group, and other liver disease-related cirrhosis. And the only blood test that is available right now for screening for liver cancer is the alpha-fetoprotein, which has a sensitivity of 40 to 60%. And 3 million in the U.S. and 486 million patients worldwide are at risk of liver cancer. So there's a huge market out there for this bioassay, biomarker. With lung, there are no blood tests right now for clinical use, and there's an estimated 200,000 new cases of lung cancer in USA in 2015. And as I mentioned before, with prostate, there is over 1 million new cases of prostate cancer projected in USA in 2015. And the only blood test available is the PSA, which has a sensitivity of 21%. And so getting a biomarker like CPE for tests to confirm it will save patients from having to do biopsies right away. So what are the milestones? Research and development milestones are completed via pilot studies, some of which I've shown you with recurrence and metastasis, and also with selected tumors such as the lung, liver, thyroid cancer. We've already published the biomarker and its performance in the Journal Clinical Investigations in 2011. And there are 2 patents filed, the method for predicting and detecting tumor metastases and the method of predicting and detecting tumor metastases in kidney cancer. And then within this patent, actually, there are a great number of cancers that have been included in there, even though it's titled in the kidney. So our plan is to continue developing the exosome CPE biomarker assay for serum and plasma from patients with different cancer types to determine reference values for normal and the disease state. So eventually somebody can walk into the clinic and we'll be able to tell them if you have potentially a tumor in your body or not. And then we will want to evaluate the cancer patient biomarker levels before and after surgery or treatment to determine the efficacy of the surgery or treatment, such as drug treatment or radiation. So this is where we're heading, and any support that we can partner with from companies outside will be greatly helpful in pushing this forward more quickly to be able to present it to patients, make it available to patients. So this is our research team. I have MDs and PhDs involved in helping with development of this biomarker. There's myself and Niev Corley, who is an excellent biochemist, Carol Patchak, who is an oncologist at NICHD. Nicoletta Lendbay is a MD that has been following a lot of patients and doing a lot of the exosome bioassays. And we have Dr. Yang, who is a molecular biologist, constantly facilitating in improving the assays and the biomarker assay itself. And we also have a number of collaborators, and this is just a few that we have done, but we continue to get people interested in wanting to give us blood samples so that we can, most of the task is really getting the blood samples from patients quickly in order to develop the assay. So we've been having help from our own NICHD people, Dr. Stephen Hewitt, Dr. Muthi, who was at the NIH in my lab before, and he's now an independent consultant. We have Professor Youngclass from Georgetown, who does thyroid cancer. And then we have our overseas collaborators in Hong Kong, Dr. Lee, Dr. Wang, and Dr. Huang in Taiwan, simply because it's a lot easier to get huge number of samples of patient tumors. And so the collaboration has been very fruitful in that direction. So I think that at this point, my seminar webinar is over, and I would greatly appreciate any questions you might have that I can assist you with. Thank you.
Great. Thank you so much, Dr. Lowe. We really appreciate your in-depth overview of today's information. I'm just going to ask Adiola to send me back the ball so I can take over the slides for a few moments. So I'm just going to remind everybody that in the chat box we are putting a link to the survey that we want you to fill out. And if you could fill it out immediately, either while you're listening to the question and answer time or while we're getting some more information over to you, that would be great, because the feedback is really critical to Dr. Lowe and to the NIH. So I'm going to ask Dr. Lowe the first couple of questions that have come in. If you have any questions, again, put them into the chat box and we'll answer them there. But the first question for you, Dr. Lowe, is, does CPE have a known biologic role in tumor proliferation?
Yes, it does. There are many papers out there that CPE can drive proliferation, and it also drives metastasis. And we have in the JCI paper that we published shown that it can drive metastasis by actually the splice variant form goes into the nucleus and binds to a protein complex in the nucleus which contain in the complex HDAC1, HDAC2, and that will then drive the expression of the metastatic genes, including NET9 is one of them, CCAM5 is another, and we have a number of genes that we know are involved in growth and metastasis that are being driven by the splice variant of the CPE.
Great, thank you. And we also have Joe Conrad on the panel now. We kind of brought him over and unmuted him. So Joe is Dr. Lowe's organizer, I guess. He's the technology transfer specialist. He's a lawyer by training with a deep understanding and knowledge of science as well. And I'm just going to ask Joe for a second to explain what a CRADA is. So there's multiple ways that people can get involved, either trying to license the technology or through a CRADA. And Joe, if you could just give us a brief overview, that would be great.
Sure, Dawn. Thanks for the introduction. Appreciate it. So, as Dawn mentioned, I'm a technology transfer specialist at the National Cancer Institute's Technology Transfer Center, and I work with Dr. Lowe as well as other scientists throughout NIH, but particularly with NICHD. So, one of the ways that the NIH moves its technology from the bench to the private sector is through what we call a CRADA, and a CRADA is an acronym for a Collaborative Research and Development Agreement. And this Collaborative Research and Development Agreement is the vehicle through which companies interact and partner with the NIH to further develop NIH technology to get it into the hands of the patient population that need it. The NIH is not in the business of manufacturing. We only develop technology, so we need to be able to partner with private industry to make that actually happen. So what the CRADA does is it gives the CRADA collaborating company the ability to work with the NIH, share the technology that's developed in the lab, and potentially license the product of the collaborative research. So the way the NIH licenses its technology is because we have to be fair. We fall under fair access laws, which means that we have to give everyone, private citizens and companies, the same access to, same opportunity to license technology, so that somebody who's starting a small startup has the same opportunity to license the technology that, say, Big Pharma does. And that's our standard licensing practice. So, but what a CRADA does is a CRADA takes the individuals who apply for the CRADA and move them to the front of the fair access line because they have a greater opportunity to actually exploit the technology. So it gives the CRADA partner the first opportunity to license the technology. We cannot promise a license outright, but what we can do is give the CRADA partner the an exclusive option to license the technology that comes out of the CRADA. And so that puts them at the front of the line in terms of deciding whether or not they'd like to license the technology. And by and large, we've never had a CRADA partner say, no, we don't want to license the technology that we helped develop. So the CRADA basically is the vehicle through which the private sector can partner with the NIH to help move that technology forward. And that's pretty much it.
Okay. Well, Joe, while I have you on the line, the questions are coming in fast and furious, which is great. I love them. Keep sending them in. But there's two questions here that I think are probably more applicable to you, and then we'll go back to questions for Dr. Lowe. So is this technology's use restricted to the USA? And the second one is, would you share raw data to volunteer organizations that will help to test the technology?
Okay, so the first question is, it's not restricted to the U.S. It's global. Whenever we file patent applications, we always file globally. So our patent applications are global. So that answers the first question. Now, can you repeat the second question for me?
Sure. I believe this person is with a volunteer organization. I guess maybe that means a not-for-profit or something like that. Would you share raw data to volunteer organizations that will help with the testing of the technology?
Yes, but that has to be done through the mechanism of some type of collaboration, be it a CRADA or a collaboration agreement. So yes, anyone can get access to it. So it's just a question of what vehicle we're going to use to make that happen.
Great. So this is a good example of where that person could follow up with me, and then we could, by sending me an email, and then we could put you in contact with Joe to make it happen. Okay, so a question here for Dr. Lowe is, Dr. Lowe, can a person just walk into your lab to get examined?
Not at this point. I think that there are legal restrictions, which I think Joe will tell you that. But if they have a tumor that is in the one of the protocols that the NIH has, then that person can join that clinical trial, and then they can give the blood, and we can do it in the lab.
Okay, great. Thanks for that answer. Another question for you, Dr. Lowe. Is the approach useful for monitoring response to neoadjuvant chemotherapy?
I would expect so. As I said, we are not there yet in terms of, monitoring people who have undergone treatment, but we can measure it in the blood right now, and we expect that there will be changes. It will drop because exosomes is a wonderful little vesicle that mirrors the parent cell. So whatever the tumor is doing in the parent cell, it spits it out in little vesicles called exosomes, and so it will reflect if the tumor is shrinking and getting better, then the vesicles that are put out by the parent cell will obviously have less of the biomarker. So in that way, we can monitor anyone that is undergoing any kind of adjuvant therapy.
Okay, great. Thank you for that answer. Another question here is, could CPE microRNA expression potentially be influenced by lifestyle modifications such as diet and exercise?
That is a messenger RNA, not a microRNA. So just a technical correction. But yeah, I'm not sure, but I think that if you don't, well, as you can see from the graph that I had, when you don't have any kinds of cancer, the levels of this biomarker is in fact extremely low. So I'm not sure that monitoring exercise would, if you don't have the biomarker, you don't have a tumor, or you have very low levels, you don't have a tumor, and therefore exercising and so on wouldn't give us a reading that would be any different from being in the control level.
Okay, great.
Does that answer the question?
If it doesn't answer the person's question, if you could just send in a follow-up, that would be great. Thanks. So I have a whole bunch more questions, but I want people, again, to be reminded to fill out the survey, first of all. And second of all, these are some things that I would like you to think about, and perhaps they will jog a few more questions from people. So let me turn to the next question for Dr. Lowe. You don't mention breast cancer in monitoring response via exosomes. Is CPE not relevant to growth and progression in breast cancer?
Oh, far from it. Because the breast cancer from the cell lines that we have done expresses, if you have a high levels of it, it'll be a very aggressive breast cancer. So while we haven't gotten a study yet done on patients from the blood assay, we expect that because the breast cancer does express this biomarker, that we will be able to measure it in blood. We just, haven't gotten a cohort of breast cancer patients yet.
Okay, great. So again,
I expect that it will be just fine for measuring breast cancer as well.
Okay. So maybe a related question as it comes to types of cancers. Is the test usable for different types of lung cancer?
We have only studied the non-small cell carcinomas, and the big study that we have done is on adenocarcinoma. It's a special type of, and the most prevalent lung cancer, and certainly mostly non-small cell carcinomas are of that type, and this is why we went for that cohort first, and also because we could get a lot of patients. There's also squamous, and we have also detected it in that type of lung cancer, but basically we haven't, of course, done every kind of lung cancer, and these two we know it's present there.
Okay, super. You may have answered a bunch of this questions. You may have covered this one already a bit, but I always ask questions anyway, just in case there's more to say.
Yes.
So what kind of cancers will the CP biomarker test be useful for in predicting recurrence or metastases?
Well, the list that we had on the slide so far, that we have done it for liver, lung, colon, thyroid, kidney. Let's see, those are the ones that we and of course the adrenal, the pheochromocytoma cancers. Because we have seen it in patient tumors for glioma as well, we think that the blood will also show up any high levels if somebody has a brain tumor, a glioma. And I think that, it's just absolutely amazing that how this biomarker seems to be elevated in so many types, and any type of cancer that often ones that we have looked at, and I've named them, we have been able to detect it. So, and also prostate, we have also seen it in prostate cancers. So it seems like some of the more common cancers that we have looked at, they all seem to express this biomarker. So I'm very hopeful, and this is why I think it's such a great screen. While if somebody goes into the clinic and they have high levels in the blood, we can't tell them exactly what kind of cancer they have, but they can then follow it up, and this is where the advantages of a general screen of this, using this biomarker will be very useful in early detection of cancer.
Great, excellent. So I'm just going to leave this slide here, and again, hopefully some people are thinking about some questions to send in based upon my 3 questions to you, like are there any additional questions or information that you'd like to have answered. But before I finish with the questions that have come in so far, I just want to put up contact information for myself. You could contact Joe, but I'm going to be keeping the stats to see how many contacts we get. So if you could contact me, and then I'll send you on to Joe, that would be great. So here's another question for Dr. Lowe. Is your approach able to test the possibility of a cancer recurrence after the person's tumor was already operated, and then after being treated by chemotherapy and then drugs?
Well, that's precisely our goal, because yes, you can detect the biomarker before the surgery, and we have, of course, patients that we have done that already, and then certainly to follow the patient after the surgery. And from the resected tumor, as I had demonstrated, we can tell you with high levels, like in the lung cancer situation, above a certain number of copies of this message of this biomarker, we can predict that there will be recurrence. And it basically, this biomarker, because we know the science behind it, is driving metastasis as well. And so when the level is high, the biomarker indicates a very aggressive tumor. And as I had demonstrated in stage 1 and stage 2 patients, they, when they have high levels, do end up getting recurrence in a very short period of time, and also, unfortunately, death as well. So it certainly can predict after the surgery what type of tumor and how aggressive your tumor was, and therefore from there predict the prognosis and recurrence. And then when the person then undergoes treatment, we can then follow it with this biomarker and see if indeed their treatment is working or not working, because the levels should be going down. And if there is recurrence, we expect it to come back up again. And if the surgery is not done for whatever reason, and is being treated, then we can also test the efficacy of the treatment by following the levels before and after treatment of the biomarker in the blood.
So, Dr. Lowe, today's presentation title was all about utilizing this biomarker in clinical trials to test the efficacy of different drugs. So I guess what you're really hoping for in that particular area, although there's lots of applications for this technology, you're hoping for maybe a company who's currently doing some clinical trials in cancer to perhaps add this into their clinical trial to help give them additional, deeper information about their drug and how patients are responding to it and the course of therapy and how it's working. So is that kind of a summary of how you're hoping to perhaps partner with a pharmaceutical or biotech company to actually test out your biomarker in a current clinical trial or one that's up and coming?
Yes, certainly that would be the goal.
Okay, and are there, another question that's come in is, are there any open clinical trials right now utilizing your this CPE technology?
Well, there are the people who are in Dr. Patchak's cohort with pheochromocytoma and paraganglioma. Those who enroll in his protocol, we are testing their, well, it's a clinical trial, I guess, to test, we're testing those patients' blood.
Okay, great, thanks. So another question here, what is the mechanism of overexpression of CPE?
Oh, when you overexpress CPE, you're driving the cells to become more aggressive and metastatic.
Okay, great. What's the turnaround time for the test?
The turnaround time is 2 days once we get the sample. So, but, you know, wherever it's coming from, the blood actually can be shipped overnight at 4 degrees. And we can, actually, we would ask the clinic or whatever to spin down the sample so that we only get the serum and they get shipped to us. Then it will take 2 days from then to isolate the exosomes and then do the measurement of the message in the exosomes. But in the resected tumors, it's the same thing. You know, if the tumor arrives, we can do it in a couple of days.
Okay, super. Another question here. If so many cancers show elevated CPE levels, can one determine the type of cancer the person has from the blood assay?
No, I mentioned that we can't do that because for that very reason. It has its pros and its cons. Its pros is that we can screen and pick up anybody, you know, that might have a cancer. Then it will need more specific follow-up tests to determine what type of cancer the person has if they are not showing any clinical symptoms. If the patient came in with a solid cyst in the ovary and they wanted to know if that is malignant, then certainly they would know that if the levels are high, it most likely would have come from the ovarian cyst.
Right, and I think maybe some oncology specialists would argue that today really cancer is a molecular disorder, and so it perhaps, you know, if we can look at it from a genetic perspective, that might be another next step rather than, you know, because as it mutates, it changes and things like that. So interesting place for it to put into the mix.
Well,
another question.
One of the things is that this biomarker is elevated in cancer cells, but other cells in the body, besides endocrine cells and brain cells, don't even make this molecule, except during development they do, and then it kind of shuts off, and then it comes back on in cancer in a big way. But not all cells in the body make this particular molecule.
Okay, well, actually, there's a great question here that kind of follows on to what you just said. So what is the specificity of the test? And if the CPE biomarker in the serum is high, can it be due to other conditions such as inflammation?
We have not tested that. That's something that, again, as in the development, that we would be collecting blood from patients with rheumatoid arthritis or one of the inflammatory diseases, and we'll certainly test that. But we haven't done any work on that yet.
Okay. Another question, is the mechanism of overexpression by gene amplification or high level of transcription or gene translocation?
There are things that we have been studying, and one of the things is that steroids, for example, they can actually drive this gene up. So again, people with high estrogen levels or progesterone levels, that would be the mechanism that it will bind to a estrogen site, a receptor binding site, and it will then trigger the gene to be highly expressed in the tumor. Is that enough? You want more science behind it?
That's enough. If they want more, they can ask another question, but thank you. Will the test method be shared so the blood may be tested on site? And I'm thinking that this is actually where you might want a diagnostic company to get involved with you in creating a kit so it could be tested on site. So I'm answering the question for you, but you go ahead and answer it if there's more to say.
Sure, it can be tested on site, but they would have to have the machine that will be able to measure the message level.
Right. So probably it's best to have a
company would have those machines, and it's easier to send it to a company to, who would like to create, to set this up as a boutique assay and provide it. It would be a lot easier rather than, it's an expensive machine to be able to measure the message at this point. No one wants to have one of those machines, in the clinic, but they could. It's not so difficult a technology to do a PCR.
Right. So this would be somewhere where someone would have to license the technology to be qualified to set up in their lab.
Sure, yeah, and there's a lot of biotechnology, small companies now that are offering boutique assays to patients who want them.
Right. Okay. Have you conducted ROC analysis, ROC analysis, to determine the PPV and NPV for your test?
We have it on tumors, yes, but not on the exosome analysis yet.
Okay. And another question here, how does this blood...
Sensitivity is actually very good. It's anything above 75%, which is better than a lot of things on the market. So meaning that if you have high levels, there's a greater than 75% chance that the test is accurate and you really have a tumor.
Okay, I'm going to go to what I know is one of your appendices for this last question. Or one of the last questions. How does this blood test compare with other cancer blood tests in terms of sensitivity and specificity? So I've put up your comparative chart here.
Okay, yeah. This comparative chart is for alpha-fetoprotein for liver cancer patients. And as you can see here, the sensitivity is much, much better. And also the other assay, which is the only one that is available in terms of a blood assay, the AFP, it cannot predict future metastasis. And also the turnaround time is a little bit longer, although maybe a day is not too big a difference. And the cost is about the same. So the great advantage is the high sensitivity of our assay and the ability to predict future metastasis and basically the aggressiveness of the tumor, which the AFP cannot do.
Okay.
Yeah.
Great.
Otherwise, the other technology is CT scans and all those things, which are very expensive. But I'm just comparing another blood test for liver that is out there, and for prostate, of course, the sensitivity is pretty poor for the PSA now, right? At 21%. So our assay would be much, much better. And I also believe that there is also no assay for glioma, and brain cancer is very difficult to detect. And so the assay would also tell somebody the efficacy of the treatment that they are getting if indeed, the biomarker is going down, that the treatment is working.
Okay, well, we're just a minute or two before the top of the hour, so last opportunity for any questions to come in. And here's one more question, but before I get to that, I'll just say thank you very much to Dr. Peng Lo for her presentation today, and thank you to the audience for all your great questions. And just a reminder, very strong reminder to everyone that in order for this to continue to be offered to everyone, we really need you to fill out that survey and give Dr. Lo and her team feedback. So Dr. Lo, this question here for you, has this test undergone regulatory review / approval, or have you consulted the device division on the analytical / clinical performance of the test?
No, we are not there yet. We need a very big number of patients to do that, and as I indicated in the slide, we are at the proof of concept and pilot stage, and there's a lot of work to be done, and if we can get some help with a partnership, that would be, we're a development laboratory, we're not a big factory here at the NIH, and so any partnering that we can do will really help us move this forward and be able to get this finally through a clear lab and licensed by the FDA.
Yeah, so I would just remind everyone that this method of working with the NIH, either through a CRADA or a collaborative agreement or a licensing agreement, is a very, very cost-effective way for you to build on solid technology that's been screened and evaluated and patented globally, as Joe said, by the NIH. So if you're looking to move your business forward or move your organization forward in this area, I would highly recommend that you contact me so we can pass that on to Joe, and you can find out more about the process and how you can get involved. So with that, I don't see any more questions, but Dr. Lo, do you want to just make some closing remarks about how you see this technology working in clinical trials and testing the efficacy of drugs as people go forward?
Well, I see it, the major goal now is to keep developing our exosome bioassay for the blood, biomarker assay for the blood, and I can see that this has got an explosive market out there, and it would certainly be very useful for following the progression of the cancer or the regression of the cancer with various kinds of treatments that the patient has been having, and also the fact that maybe the drug is not working, that they will know very quickly that from the blood assay that maybe the drug is not working and they need to switch therapies. So I see this as a very exciting area, and the whole area of exosome technology is very exciting now to be able to measure a messenger RNA, basically, which is the biomarker we're measuring in the exosomes for the future. So.
Great. Well, thank you so much, Dr. Lo, and thank you to Joe as well. And we look forward to having even more science come this way, and more people around the world learning about the great opportunities to partner with the NIH and help move science out of the NIH and into the real translational element of patient care as fast as humanly possible. So thanks again to Dr. Lo and Joe, and thank you for everyone who attended today. We look forward to hearing from you and having you participate in the future. And don't forget those surveys. Take care. Bye now.